Okay, thank you very much for the invitation and for introducing me. So as Sergey said, I will be today talking about mostly about polymers, which inherently has a diagnostic modality and how we combine them with how we integrate them into biomaterials for medical applications. So first of all, I would like to give you a little overview of materials which I will be talking about today. So I will be talking about polymeric nanoparticles. I will be talking about thermogelling polymers and injectable thermoresponsive polymers. And then I will also talk about the most recent studies I did, and those are focused on biomaterials, which could be used then for tissue engineering and related therapies. So here is a little overview of why is it important to be able to track these materials in vivo. So for example, for the nanoparticles, it's useful to be able to monitor how these materials, if these materials accumulate in some specific tissue of interest, and then you can monitor therapeutic response of the system and you can link it to a presence or the absence of the nanocarrier, and that is actually a special class of diagnostics. It can also be used for tissue labeling and cell labeling for cell-related therapies. For the thermogelling polymers and injectable thermoresponsive polymers, we are able to monitor the dissolution rates, and that we could later link to drug release rates and then be able to adapt that. And then for the biomaterials, it's for tissue engineering, one of the parameters which is very important for us is, of course, the degradation rates, the degradation kinetics. It's actually a parameter which, let's say, determine if the tissue engineering will be successful or not. So it's very important. We also implement a lot of 3D printing methods to get a specific shape for the biomaterial we are designing, and it's very important for us to know if this shape is maintained when we implant it, and how does that what's gonna be the effect on the tissue engineering in future. So today I will be talking about two diagnostic techniques, but mostly about fluorine magnetic resonance imaging. This method is not in clinics yet, but I think it will be introduced very soon. It has high potential because of its biocompatibility. It's a complementary method to the classical magnetic resonance imaging, where you obtain the anatomical information about about the body. And then when you measure the fluorine, if you have a fluorine measuring coil implemented in your MRI, you get the exact location of your scaffold. And then when you combine it, you get all the information needed of your biomaterial. So for this specific method, we are using a fluorinated polymer, polydifluoroetheracrylamide. This polymer is in fact thermoresponsive, and that's very convenient because theoretically you could replace the commonly used thermoresponsive polymers like NIPAM with this polymer, and you would have the advantage of having the diagnostic modality already incorporated in the materials. So the next technique I will also be talking about today is common radiology, so the CT, micro CT, or X-ray. For this method, we recently developed a polymer which has this specific motif. That is a motif which you can find in most of the common contrast agents which are used for radiology and which are FDA approved. And we transform this monomer into a metacrylamide, so that this is how we integrate it in the biomaterials, which I will be talking about later. So first of all, I will start with very brief overview of old projects which I did during my PhD, which I did on this specific fluorinated tracer. And I will try to share with you only the most interesting results which we found out. So we made nanoparticles based on this polymer. We made these nanoparticles to disassembly upon a specific stimuli. So we have drug delivery systems, and then we also developed injectable implants. So the first interesting results we obtained with this polymer is that when we made block copolymers, we obtained this special type of nanoparticles, which we described as physically cross-linked nanogels. And these particles, the polymer is homogeneously spread it over inside the core of the particle. The particle has high amount of water. It has hydrophilic corona, and we also describe that it is actually in equilibrium with some smaller micelles. We think that the reason why is this is because we have this hydrogen bonding in the extra hydrogen bonding on the difluoromethylene group, which is in the polymer. Another interesting result we obtained was when we made statistical copolymers of this fluorine emerald tracer again. We combine it with the monomer which has imidazole in its structure. Imidazole can be protonated below pH 6.5, I believe, or 6. So this can be used that when this polymer is protonated, or when this polymer is in pH below 6, the imidazole is protonated, and that helps for the polymer to be fully dissoluble. So they can then you can inject it in the lower pH in the body, or in the body, and then when the material is buffered to pH 7.4, you should obtain a precipitated polymeric depot. We also integrated this hydrophilic monomer. The monomer was integrated in order to be able to adjust the water solubility of the polymers, and the idea was that the more of the hydrophilic monomer we will add, the faster these polymers will be dissolved from the place where they were injected. And this is actually something which works. We were able to monitor this with the fluorine MRI. And the interesting result I wanted to share with you is that when you look at this polymer, specific one, the cloud point temperature of that polymer is way, way higher than the physiological temperature. That means that when you inject it, it should be fully soluble in water. So we were actually expecting that this polymer will go away or like wash away immediately after the injection. So we use it as a control. But the interesting thing is that we found out that we can still see it after almost 2 months. The half-life of the depot is 31 days. So we propose that there can be some interaction of the polymer with the tissue. It doesn't really matter if it's in precipitated depot or if it's fully dissolved. There has to be some interaction with the tissue. So to prove that, we made 4 homopolymers, 4 thermoresponsive homopolymers. We modify them with the fluorescent dye so we can monitor optical imaging. We injected them, and again, we were monitoring the dissolution rate. The interesting part is, again, that we included a polymer which has LCST around 50 degrees Celsius, so it's again above the body temperature, and that polymer we were able to see, still 1 month after the injection. So we proved that this is actually the case. So we were monitoring if the interaction with the tissue could be internalization into the cells. And we showed that these polymers actually goes into cells very quickly. You can see the polymer here. The polymer is in green. You can also see that the polymer, it has some sort of grainy structure. So we think that the polymer is inside of some sort of vesicular structure of the cells. So this could be actually very interesting for using this polymer for long-term labeling of cells. And also it could be used for possibly for drug delivery into the cells. So now I will move on to a bit more recent projects. Recently, I made nanoparticles, which can physically cross-link into hydrogels. It was already described here as well today. And then I made chemically cross-linked materials, which could be used for tissue engineering. So let me start with the nanoparticles. I made 3 block copolymers with the fluorinated thermoresponsive polymer, and I used 2 different hydrophilic polymers. I will be only talking today about the polyethylene glycol. These polymers, the specific architecture is because, which was already said today, that these polymers make nanoparticles in low concentrations. And then when you increase the concentration, you create these bridges between the nanoparticles, and the nanoparticles can create these self-assembled, physically cross-linked hydrogels. So I made two groups of polymers, one which has a very long polyethylene glycol and very short thermoresponsive side chains, and then inverse polymers with short pack and long thermoresponsive side chains. You can already see that these two groups give you completely different nanoparticles. The first group gave us, again, the physically cross-linked nanogels, the particles which I was talking about in the beginning of this presentation. And then the second group gave us these worm-like structures or assembling, aggregating micelles. And when we actually heat up this sample above the LCST, you can see here, we already saw in the TEM that these polymers create these like monolayers of particles, which could already give us a hint that this system will actually be able to make the physically cross-linked hydrogels. So we only investigate later these three polymers. Here you have a little overview of the LCST profiles of or the cloud point temperatures for these three polymers. So let me just go briefly through that. At the low concentrations, below the cloud point temperatures, you will have nanoparticles for the system. And then when you get above the cloud point temperature, the samples, the particles precipitate out from the solution. And then when you increase the concentration, the precipitate can attach to each other and aggregate and create physically cross-linked hydrogel. And then when you look at the physiological temperatures, you can see that all three polymers are able to make nanoparticles in low concentrations, hydrogels in high concentrations. And also you have the advantage of having the diagnostic modality. So these polymers actually can cover quite a range of possible biomedical applications. So now I will move to my most recent projects, which are focused on chemically cross-linked materials for tissue engineering. In tissue engineering or regenerative medicine, the idea is that you harvest patient-derived cells or patient-derived stem cells, you seed them on your biomaterial scaffold, then you pre-grow the tissue, and then you implant it back to the patient. And then when it's in the patient, the material should degrade and should be replaced by the tissue. So you can see that degradation, determining the degradation rates is really crucial for these applications. And the degradation rate should also match the growth of the rate of the growth of the tissue. So if there is a mismatch, it will be a problem. And second, what I will also talk about is that it's important to have a specific mechanical properties for the biomaterial scaffolds. It should always match the mechanical properties of the tissue you want to regenerate. And if there is a mismatch, the cells will not grow on that material. So the current approach for these materials is, for most of the studies, is that the materials, when they're implanted, when they want to measure the degradation rates, or they want to see what's happening with these materials in vivo, they take a large group of animals, they sacrifice them in specific time points, then they dissect the material out, and then they obtain a relatively limited amount of data. So of course, it would be really ideal to be able to monitor this non-destructively in vivo. So the first two projects I will be talking about are based on gelatin. Gelatin comes from collagen fibers, and those comes from extracellular matrix. So gelatin is inherently cell interactive and is used a lot in the regenerative medicine, for the regenerative medicine application. We modified it with photocross-linkable groups. So after the photocross-linking, you can obtain your hydrogels, and it can also be used for the 3D light-based 3D printing methods to obtain your 3D printed structures. The most common materials, the most common material based on gelatin is so-called GELMA, where we modify the amine groups and we transform them into metacrylamides. We also recently started to use another material which is called GELMA-AIMA, where we transform the carboxylic acids into metacrylates. So then when you cross-link this material, you obtain your hybrid structure between the gelatin and polymetacrylate or polymetacrylamide. So the first idea we had, which was the most simple way how to introduce the diagnostic modality into these structures, was to just add it during the cross-linking. So then you obtain these hybrid structures of gelatin and the synthetic polymer. For the first project I made, I used the GELMA with different degree of substitution of amines, 30, 70, and 100% of amines were modified. And like this, you can kind of play with the degradation rates and the cross-link density of the material. So here what you can see that when I was adding the co-cross-linker to the gelatin solution, I kind of artificially increased the concentration of the hydrogel. It was the first approach, but what we saw later is that when you, for example, look at the mechanical properties, these three materials are controls, and these three materials has the monomer in it. You can see that we are kind of making the mechanical properties significantly stronger, or almost twice stronger. We are making almost twice stronger material, and we are also decreasing the swelling ratio. So this is introducing some tunability, but also something we didn't really want it in the beginning. We wanted to add the diagnostic modality without really changing the properties, and I will show you later how we solved it. So these materials, we wanted to see if we can imagine them in vivo, and if we can actually determine the degradation rates. And for that purpose, we 3D print porous scaffolds, and I chose this very simple 3D printing method, which actually anybody can do in their lab. So you use so-called sacrificial mold. So you 3D print your scaffold, then you submerge it in the cross-linking ink, then you cross-link it, and then you can dissolve the sacrificial mold to obtain your hydrogel. So this is very simple way and very fast way how to prepare porous hydrogels without any need for optimization of the 3D printing. So we implanted these materials in vivo. We also implanted a control. We could somehow, without the monomer, we could somehow see it in the MRI in the beginning because it was porous, but at one point we couldn't see it anymore when the pores were gone. So, and you can see that the scaffolds with the fluorine are very nicely visible. So we were able to determine the volume and the location. So what I said in the beginning is that we significantly affected the properties of the hydrogels. And here we also see that when we were measuring the degradation in time, this is measured within 1 year, we saw that 2 of our materials are not degradable actually, but the cross-link gelatin normally degrades within usually 3 months. So we significantly affected the degradation. Here we can see that one of the material degraded within 1 year. So we kind of still have a little window of tunability, but this is not really something we wanted in the first place. So in the next project, I will not go into details. I will just show the basic concept, is that we decided that because we know that the degradation can be affected highly with the concentration of gelatin. So when we're adding the co-cross-linker, we were diluting the gelatin amount to keep the material content the same. And we found out actually that the materials has comparable mechanical properties and as a control, and they are also biodegradable that we so far only tested in vitro. So this is really nice result for us. And in the next project, we did the same with the contrast agent for radiology, which I was talking about in the beginning, exactly the same way. Here we employed the GELMA and GELMA-AIMA. So just briefly, I will show you how it looks like when you add the contrast agent. Here are 2 control groups. It's a crushed hydrogel in PBS. And then the more contrast agent you add, the more of the contrast you obtain, obviously. And then we compare it to the tissues or the radio density of the tissues. and we found out that our materials has the same radio density as bones. So that means that they should be visible very nicely in vivo. And then we decided that the materials with the lowest amount of contrast agent, we will implement we will implant and we will measure the degradation kinetics. So we implanted them two different ways. Again, we used the porous scaffolds for this study. And the second approach we use is so-called injectables, where you take a hydrogel, you dry it, then you blend it, and then you reswell it and inject. And the particles have some specific gaps between each other, and that helps for tissue engineering that the cells can nicely migrate through these pores. So it's actually behaving similarly as a porous scaffold. So here briefly about the degradation. So thanks to the fact that the radiology has very nice resolution and you can obtain all kinds of data from it, we were able to actually determine the mechanism of the degradation of these scaffolds. So when you look at, for example, the volume, you can see that in the beginning, the material swells. So that is probably because you have a bulk degradation. So some cross-linking points are cut and then your material starts swelling. And at one point, the material volume starts rapidly decreasing and the materials actually degrade within the 3 months. So this is something nice that we found out that with the contrast agent, because we're adding very little, the material actually behaves very similar, almost the same as gelatin without the co-cross linker. So that's an advantage. So another thing what we looked at is the average radiodensity, and you could see that in the beginning, at the end, it's almost the same. It's fluctuates, but it's almost the same, within the same range. So we concluded that this degradation is actually surface degradation because the material is released from the surface, not within the material. Otherwise, the radiodensity would decrease. And also, last point is that when we use the Gelma AIMA, the highest cross-linking density material, again, we had a material which has no degradation. But this could be, for example, used for gelatin coatings, which normally degrades very fast, but now you could have prolonged degradation. And at last, I will, if I still have 1 minute, recently we made also materials based on polycaprolactone cross-link scaffolds. Those are not, because polycaprolactone is hydrophobic, so it's not creating hydrogels, but it's creating compact hard scaffolds. These scaffolds, if you look at, for example, here, are known for having very strong mechanical properties between tens and hundreds megapascals is the Young's modulus. And that could be actually used for tissue engineering of bones. So because it's also very important to be able to monitor these materials in vivo, we decided to implement again the contrast agent into these materials. And the most important thing we were watching is that it doesn't affect the mechanical properties, because it's really important for us that the material maintain its strength. So what we could see that with this relatively minute amount of contrast agent we added, the mechanical properties were not affected, so that was really nice. What was affected was a degradation. Here you can see degradation in vitro. But yeah, first I will walk you through this. So the black and gray is a control group. You could see that in half a year it doesn't degrade at all. And then, for example, these two are the highest amount of the co-cross linker, the hydrophilic contrast agent, and those are degrading fully in that half a year. So the thing about the cross-link PCL is usually that it degrades too slow. So it's actually very convenient for us that we are able to actually also tune the degradation or accelerate the degradation of the material. So that was really nice. We implanted the material in one mouse just to see how it behaves. And you can see that the radiodensity of these materials goes way higher than actually necessary. So we only implanted this material, which is like the lowest amount of contrast agent in it. It has approximately 5% of iodine. And we were able to check how this material behaves in vivo, and you can see that after half a year, it degraded to 40% of its volume. So this is really nice. We could actually show that indeed it degrades in vivo. And now we are doing a bit larger in vivo study, but I will not really go further because it's not concluded yet. I would like to summarize it. I will just say that the materials are biocompatible and well tolerated. We also don't see any accumulation anywhere in the organism, which is very nice. And the materials which are supposed to be cell interactive, they were cell interactive, so that's very nice. To thank you for the attention, and I would also like to invite you for a conference which will be next year in Antwerp, which is organized by European Biomaterials.